Okay, hello everyone. Good day. Welcome to BioRTC webinar series. This is the second webinar series brought to you by BioRTC Biomedical Science Research and Training Center here from Yobe State University, Damaturu. And for our host, for today's host is going to be Dr. Murtala. Dr. Murtala is currently a research fellow at BioRTC. He's a staff of at Adwa University, Katsina. He has done a PhD in biochemistry from University of KwaZulu-Natal, South Africa. This is a brief introduction of Dr. Murtala, but we're expected to hear from Dr. Murtala as he's seated and sits to give the talk. Thank you, Dr. Murtala. You're welcome.
Thank you, Kundi. I think I would start sharing my screen then. Okay, I hope you can see my screen.
Yes, all good.
Okay, thank you very much, Kundi, and everyone joining us today for this webinar. I have to maybe sincerely apologize for last time. You know, sometimes every time you plan one thing and then at the time when we were about to start it, then we encountered some technical problems that necessitated that we shifted this webinar. But today I think we are good to go. My topic is how to design effective research projects and utilize molecular biology and microscopy tools in your research, a comprehensive guide to BioRTC. I think when I was designing my presentation, I made it in such a way that it will benefit students who are willing to come to BioRTC to conduct their research, and not necessarily if they are going to utilize molecular biology or microscopy tools. If you are going to use any of the facility available in BioRTC, I believe the webinar is going to be useful because it's going to be a guide, a comprehensive guide to what you do if you want to visit the facility. As a way of introducing why there was the need to establish a center like BioRTC, in 2021, some researchers of the center conducted a comprehensive research on the research capacity in Africa on neuroscience research. And you can see that we already know that the problem of research in Africa includes and not limited to low funding, low expertise, and poor infrastructure. And according to many reports, Africa produces just about 15% of global research, but the global burden of disease, Africa bears more than 25%. So there is this disparity about what Africa is doing in terms of research and what it should be doing in terms of what is affecting it. And a closer look in this paper, for example, shows that not only Africa is producing research quite all right, but the quality of the research is very, very low. If you look at the advanced techniques that are produced in African countries compared to other countries like Brazil, Australia, Japan, UK, and the US, or generally the global north, then you see that Africa is still lagging behind in terms of the quality of research. The papers are there, but the quality of research is low. And this is like transgenic models also is almost non-existent in Africa in terms of neuroscience research. So this cuts across other areas of research as well, meaning that if you go to biomedical science, all biomedical science disciplines, even engineering is even worse there. So even social sciences. So this problem of research that is lingering in Africa is already there. So that inspired a group of a lot of people, a lot of scientists, especially those that come from Africa. And one of them is none other than Professor Mahmoud. He founded BioRTC in 2020 in collaboration with the Yobe State University, who was the home for the center, then including other collaborators like Trend in Africa, and then there are a lot of like a consortium of collaborators that come to see that BioRTC come into being, that like the equipment come from different parts of the world, especially Europe. University of Sussex is one of the active collaborators giving equipment to the center. And then we received other equipment, say, from other collaborators in Spain. So this equipment will be received, and then the Yobe State government is responsible, and through Yobe State University is responsible for accommodating and ensuring that the equipment are installed and in good use. And then we have the equipment that are there, and which I'm going to show you when I go to my next part of my slide of my presentation. And then the facility, there's a lot of facilities in BioRTC, but the one that is more, I think, we are most known with is the imaging facility that is there. We have currently the only confocal microscope in Nigeria, and that is something that I'm going to talk about later. The center also will be responsible for conducting research. I'm going to talk about the kind of research that are performed in the center. Then the center also will be involved in training young and upcoming biomedical scientists within Nigeria and West Africa in general. And this includes, since the inception of the center, a lot of activities held. In 2021, there was a symposium that happened. Then in 2022, the infamous summer school on neuroscience, open source hardware, and clinical collaborative science symposium and bioimaging. Then in 2022 as well, October 2022, there was another workshop for clinicians on data analysis. Then in 2023, we had another workshop on confocal microscopy. So there is a lot of activity happening in the center. So the kind of research that I've performed, that a lot of research can go on in BioRTC depending on who is coming to do the research. But the ones that I'm showing now are those that are conducted by the PIs on the center. These include Dr. Mahmoud himself, who is involved in the research on neurological disorders. Then Professor Raouf, who is currently in France, manages his own laboratory. He's working on optical method for behavioral neuroscience research in Drosophila. Then Dr. Takeshi, who is now in the US, is also working on non-model organisms to study specific adaptations of the nervous system. So these are some of the research that are actively happening in the center. So I think now I will go to the website. How do you access biochemistry? We do have a website, and all the information you need on BioRTC are available in the website. So let me go to. Okay, so the website www.biortc.com will take you to the homepage, where you'll see that, of course, the information you can read about RTC, what it is and what it does. Then we do have some gallery on our past events, which you can look and see what. But I think the most important thing that you can find is the resource that is available. I think that is where, when I was there, when people try to ask me about what they can do in the center, I always refer them back to the website, because all the equipment that are in the center are already listed in the website. So you can come and click on the equipment. Then we have all the list, and we categorize them by the category of. So if you have already designed your research and you want to come to BioRTC, I think the website is a good way to go and see what kind of equipment are there, what can you use to what equipment is available for you to do. For example, the animal models that are available in the center now are Drosophila. We have wild type and the transgenic Drosophila. Then we have rodent models, which you can come to the center and work with. In terms of microscopy, we have different assorted microscopes. And the one, I think the most popular one now is the Zeiss LSM 700 laser confocal microscope. I think, if I'm not mistaken, 2 more are coming towards the end of the year. Then you have the Zeiss Axiovat inverted fluorescence microscope and a lot of dissecting microscope that you can use for your research. Then you have DNA and protein electrophoresis equipment and imaging systems. Especially this LICO Odyssey gel dock system, I think is one thing that I need people to really try to explore, because it's a very, very powerful machine. With proper research design, you can answer a lot of research questions with it. It does fluorescence. If you have fluorescent antibodies for your Western blots, for example, or you do agrostil electrophoresis, you can do quantitatively and very, very accurately do chemiluminescence or fluorescence-based detection methods. And So that's something that I think is one of the equipment that are very, very underutilized, because it's the only one that I can think of very available in Nigeria, and it has some powers that other gel documentation systems do not have. So I think it's something that people should really pay attention to. Then a lot of PCR systems, you have thermocyclers, normal PCR, and for qPCR systems. Cell culture facility, if you're working with cells and you need a cell culture facility, I'm going to talk about what are the procedures of accessing all this. So, but these are some of the facilities that are available for you to access. Then, of course, in terms of other equipments like Luminex multiplexing system, I think it's also a machine that is highly underutilized, a very, very powerful machine that is highly underutilized. So think about when you are designing your research, what application do you have for Luminex multiplexing systems? We have the FMAX, which is a microplate reader that does fluorescence, another powerful machine. Then we have, of course, for tissue sectioning, or a comprehensive collection of tissue sectioning machines that you can come with from your research. You can come with all your say chemicals, and with your tissues you should be able to perform your tissue processing and sectioning and staining and imaging in the center. The website also contains information about the people in the center. I think one thing that I need to call attention is the faculty that are available. So I'm going to talk about, when I come to research design, I will talk about what you can do if you are new into research and you want to start your research. Sometimes somebody is already working in the field and is already affiliated with the center. So you may consider contacting them and making a kind of collaboration with them, and your work should be easier to perform because they should have all the reagents and the equipment you need, and they should be able to guide you more compared to when you are coming with a research that nobody has information about. So Dr. Mahmoud already has been introduced, and we have Dr. Takeshi, Dr. Raouf, and Dr. Rainey, Dr. Babawaru, who is the current CMD of Yobe State University Teaching Hospital, and our open science coordinator and principal investigator, then Dr. Andre. So these are some of the people that are involved, and you are free to look at their profiles, see what they are doing in terms of research. And if your research objectives align with theirs, then you can contact them directly. Their email should be available. Contact them directly, and you can initiate a form of collaboration, and you can come to the center and do your work. There are many other resources, like you can see our advisory boards and what we have done at the back. So these are some of the things that you see how the center, the story of the center and what it's doing globally. So you can also look at our previous what we have done so far, like in 2022, what has been done, in 2023, what is coming up. If there is, for example, currently now we are receiving applications for the summer school of 2023. So the application form is accessible through the website. And so I think this is a brief overview of the website. And if you are welcome to go there and browse it and see what information is useful for you, but you should be able to have a lot of information on it. So what kind of experiments? I think this is another question that I've been asked during my time in BioRTC. What kind of experiments can I do in the center when I'm there? So this is not an exhaustive list, but these are some of the common experiments that link up or that merge expertise from different fields, or different students or different people coming from different fields can utilize. Like fluorescent microscopy is one of the areas that BioRTC really has the capacity to change Nigerian and African science, because of the presence of all those microscopes that are not found elsewhere. So think about, even if you have not planned to include microscopy in your research, think about incorporating fluorescence microscopy. It's a very powerful technique. You can answer many research questions using it. Then, of course, agarose gel electrophoresis, PCR and qPCR, SDS pH electrophoresis, Western blot, ELISA. All these are some of the techniques that fully now, if you are to come to BioRTC, I'm going to tell you the caveat when you come. There's some conditions, of course, and some of the requirements to run it. But This technically you should be able to run these assays in BioRTC. So I think before I begin about designing your own research, I would have to talk about the scientific method, which I'm sure a lot of us are familiar with. But just to refresh our memory, the scientific method is a systemic way through which scientists ask questions and come up with data to answer those questions. So for example, before you start, or definitely you must have selected a research field, and then you should have a question that you are targeting that will lead you to design your experiments and then test those experiments, test with these experiments. Then you have to ask yourself some of the question that are these experiments working the way you expect them to? If they are working, then definitely you now have the data to analyze and draw conclusions and report your findings. And sometimes this is something that I think a lot of new researchers don't realize, is that sometimes if you design your experiment, it doesn't necessarily mean that you are just going to do it once. Sometimes you need to it's a iterative method where you go on and do the experiment and they are not working, or the question you ask is not answered. You have to go back and troubleshoot the procedures, carefully check what is happening, then you come back and test with experiments. So in this method, in all these research pipeline, where does BioRTC chip in terms of improving research? And this is where BioRTC, the area highlighted in green, is where BioRTC basically is very, very handful when it comes to facilitating your research. That is where, at the point where you start your experiment, is where you should be thinking about incorporating BioRTC in your research. But like I said, the research design involves a lot of things that I'm hoping that anybody who is listening is familiar with. But one thing that I notice is that sometimes for the people that want to come to BioRTC, they are really, really in very, very early stage of their research. For example, they just know that they are going to work in a particular area, but not necessarily having the design the experiments, and that really makes it a little bit difficult sometimes to access the facility. Because think about it, anybody who is a biomedical scientist and wants to work with all those experiments that I mentioned can come to BioRTC. So really there is not enough time for the staff in the center to start designing the work for you. So designing the work involves you going and knowing the exact methodology that you are going to use. You have already know the reagents, the details of the reagents you are going to use, the details of the equipment you are going to use. If there is no this equipment, what other alternative equipment is available for you. These are all the things that you should do during the design process. So there is no harm when you have already know the questions that you are going to ask. I'm going to demonstrate how to ask the right question in research, but suppose you already have the question. There's no harm if during the design of the experiment you initiate contact with the center, and the people in the center can tell you this is what we have. If you, for example, you want to do one experiment and then the center doesn't have the equipment, we may have another equipment that should be able to substitute what you already proposed. So then you can, during your design, you can substitute with what is available in the center. So I think this is something that if for those intending to come to the center, if you do not have that, this is something you should start thinking about. Just because you have a research topic doesn't mean that you are ready to start experiments. You need to ensure that you have a comprehensive idea about what you want to do, down to the last reagent that you need. Because sometimes, like I told you, experiments are very, very difficult to optimize sometimes, and sometimes it's labor and cost intensive, that you don't want to spend a lot of time, a lot of resources, a lot of funds in the initial stages of your experiment, just for you to come and realize that you can't finish the experiment because there isn't one reagent or another. I will give an example when I come to an actual research design that I'm going to give you as an example. So this is something that I really need us to pay attention to, intending visitors to BioRTC to notice, that you need to have a comprehensive research design with all the reagents and everything you need before you initiate contact. Like I said, there's no harm if you during the design process, you initiate contact so that you can optimize, but definitely you want to come to the center and start your experiment when you already have design and pin down exactly what methods you want to use. So I categorize this cartoon that I have is my way of trying to explain to especially young researchers what is happening when you are designing your research. So we have two categories. There is a hybrid, but I can summarize all those that are starting research into two. There is group A, who are the researchers that are in a lab that already has all the methodologies, all the design. They have asked all the questions that you need. You are just coming there as a student and they are guiding you on how to answer the questions. You are just taking a part of the research to do. So for those kind of students, you will find that the starting point has already been defined for you. And sometimes you're just a sort of kind of going to climb down to when you do go for your data collection. That is majority of those students, majority of students in especially in Nigeria will fall, I believe, from what I've seen during my time by RTC, will fall into category B, which are those students that have no idea where to even begin. So if you are in category B, then you need to understand that you really have a lot of work to do. Like I'm going to demonstrate in my subsequent slides, that you need to really do a lot of work to come up with a good research design and good strategy to go into your research. For those of us that are lucky and they found themselves in group category A, usually you will see that by the time you go to the lab, they already have all the reagents that you need. They already have all the models. If you are using model organisms, for example, that you need, all you need to do is to ask your question that is specific for you, and you can use the facility there to start answering the question. So I'm going to focus more on, and imagine that majority of, this is just my guess, that those that are listening will fall, a lot of them will fall into, and especially those that want to come to BioRTC will fall into category B. This is where people that don't even have, they know that, for example, the supervisor knows, they know that the supervisor is working in neuroscience or in malaria. You know you are going to work on that area, but you don't really know exactly where you are going to start. So I'm going to give you some tips when you are designing your research projects. What are some of the tips that you want to consider when designing your project? One of the number one is you need to really, this is something especially biomedical scientists are called really not doing. You should have a research question. A research question may not necessarily be ending with a question mark. You may not have a question the way, especially if you have been involved in social sciences. Usually those, they usually, I think, more into, or their research involves more asking the questions in terms of a clearly in a question format. But in biomedical research, sometimes the question may not necessarily be in form of question, but you need to know exactly what you are trying to answer. If I'm not going to go into the details of that, but I think if for all of us that did research methodology, you know that in biomedical sciences, sometimes the research question may not necessarily be ending with a question mark, if I may put it that way. So they should be specific, measurable, and achievable. So these are all your research questions should be definitely specific, measurable, and achievable. So before you start, whenever you have narrowed down to where you want to do, you must formulate a research question. That will definitely immensely help you in designing the experiments you want to do, because your aims and objectives will align to your research question, and those aims and objectives are what you are going to be designing the experiments to answer. You must ensure that also you choose the appropriate research design. So ensure that the design of the work you want to do is appropriate for the research question that you want to answer. So just because someone has used SDS page electrophoresis in their research and you think that this is something you can do in BioRTC doesn't necessarily mean that that is the most appropriate experiment to answer your research question. So sometimes microscopy is best for you, sometimes it's Western blot, sometimes some other form of experiment. So you need to know that the experiments you are going to do is appropriate for the design, for the experimental design you adopt. Also, select your sample. This is if, for example, those that are going to the fields, ensure that you, before you begin, you have appropriate sample size, and ensure that will ensure that the results that you are going to get are statistically significant. So sometimes somebody will come with just maybe 2 slides and this is what I want to image just to get a good picture and go and put it in the thesis. But sometimes that's not the most appropriate way of doing it. You ensure that if you are working with animals, ensure that the grouping you are going to do covers your control positive, both positive and negative controls. And maybe if you are doing a dose response kind of experiment, ensure that the doses you are using are appropriate, ensure that the interval you are giving, the number of groups you are including are appropriate. Because even, like I said, these experiments that you are doing are going to be labor and cost intensive. So you must ensure that what you are doing will be useful. And for research to be useful, it has to be statistically significant, or what I'm saying, statistically accurate should have been the right word there, should be statistically accurate, because some of the findings may not necessarily be statistically significant. So the one that that's relevant to my presentation, most relevant to my presentation today, is that you must ensure that you develop a detailed experimental protocol, and your experimental protocol should outline all the steps involved in your experiment, including the materials, the equipment needed, and the procedures to be followed, and measures to be taken, everything, how you are going to prevent the occurrence of errors, and everything that you need in terms of the experiment, you need to do that. This is all part of the research design. And then test your experimental procedures. Well, sometimes when you come to the lab, it's not every time that you work once and everything is working the way you want it to. So that is for those that will be contacting BioRTC, the people in BioRTC that I'm going to send samples. Ensure that you are not sending your precious sample that you spent 2 years to get, and you send all of it to BioRTC for measurement for the first time. Sometimes the first time doesn't work. Sometimes the procedure, one of the buffers is not working. Sometimes some of the chemicals are not working. Sometimes you need to optimize. So these are some of the things that during your design you need to be aware of. That not every time you do experiment that you're going to get it the first time. So sometimes you need to really test your experimental procedures. So in this context, before you, depending on the type of experiment you are going to do, plan your visit in 2 ways. That the first time you are coming, you are solely going to test that your procedures are working. So I have, for example, some people that want to test for, do a Western blot and measure the activity or the expression of caspase proteins. So caspases are some proteins involved in apoptosis, for example. So they want to measure them using Western blot. Sometimes it may not be wise for you to run the experiments with all your groups and just bring the samples that I want to run Western blot and homogenize them and just take all of them and run SDS page and just blot with your precious antibodies. Sometimes it's better if you go and take normal rat, which should have a baseline expression of the protein you are targeting, then take one that actually reduces the number of animals. There's this three R's in research, that is reduce, replace, and refine. So you want to reduce the number of animals you use. So there's no point in you going and do experiment with 30 animals and the procedure doesn't work. Then you kill 30 animals for no reason. So it's better to come and you take one rat, take its liver or brain or whatever organ, test that actually the chemicals and the reagents you have actually do work to detect caspase or any other protein. If you see it, now you know that when you are coming to compare expression level in different animals, then you know that you are going to end up with a positive result. So when you are coming to, my advice is that if you are coming to conduct experiment in BioRTC, plan it in 2 ways. One, the first time you'll be coming should be a short stay where you come and optimize the methods you are going to use. And then the second time is where you have gone and done your experiment, or whether you are bringing organs or you are coming to BioRTC to work with the animals that are available in BioRTC, then you can come and spend more time and you know that whatever you see is going to be the data that you are going to end up using in your thesis. So this is some advice, a tip that I think it should be everybody that is intending to come should consider. And then control for extraneous variables. This should be in form of controls, so ensure that you have appropriate controls. Sometimes you have positive control, negative control, drug control. These are all the things that will ensure that you minimize the errors that you are going to encounter. You also minimize the cost and the labor that's going to incur during your research. So I give an example of, if, for example, you want to design a research starting from a research field of malaria, for example. Now, I'm giving an example, but if this is applicable to any area of research that you want to do, before you start the research, of course, you know the research field that you are intending to work in. This can be malaria, this can be neurodegenerative disorders, this can be diabetes, this can be hypertension, any of the research fields or metabolic disorders in general. You know, before you even the moment you are admitted, the moment you intend that you are going to come back for master's or PhD, or if you are a researcher, you already know the research field you are going to work with. So that's something that we all know. So for example, you narrow down to the area. You need to narrow down to what one area of research I want to work on, the epidemiology of malaria. So what aspect? So I can begin as a global topic that I want to investigate the prevalence of malaria among under 5 children in Yobe State, for example, right? So before I can design an appropriate epidemiological study on malaria, I need to follow this reiterative stage of going and reading the literature, coming back to analyze what I read, and then taking notes, and going back and analyzing and taking notes. This will, at the moment you start going round and round, and this can take months to do. Sometimes designing a design an experiment, it takes a good part of a good research. And especially if you are going to sometimes some research start with designing the research, then seeking for research grants. So when you are seeking for research grants, sometimes you need to spend a long time. What you are doing is reading, analyzing, and taking notes and designing the work. So what you can do, what you can see is that the moment you do this reading, analysis, and note over and over again, you may find that for me to conduct a prevalence study on malaria, these are some of the things that I need, that I know that I'll discover that I need an ethical clearance. For example, I will need a consent from the patient that I'm going to deal with. This is just an example. Of course, I will know that I may want to go to the field. This may be an area you want to start with. Are you going to the field to collect the samples yourself? Or is there a biobank that have already been collecting malaria samples over a long period of time, or you want to go and analyze the biobank? So these are all the things that will come up during your reading and analysis and taking note stage, and this is all design at the design stage. So you would think that the things that I wrote there, the ethics clearance, the consent, and the sample collection, these are some of the subtle things that sometimes if you have not gone into the literature to see, you may not realize that I need them until the time you come to start your work, and then you realize that I really need these things. So these are some of the things that you will discover that I need this thing. So you discover that, okay, I need sample to collect whole blood samples. So what can I do to diagnose malaria? Because I'm going to know what is the level of malaria amongst the population that I chose. So I can use many, many ways. One of the ways that I can use for the diagnosis is to use microscopy. This is something I will discover during my reading and analysis stage. I will find out that I can use microscopy, I can use RDT, I can use, or majority of the literature that I read have used either RDT, microscopy, or PCR, or sometimes even a combination of all the three. So now I would decide which one I want to use. So this will depend on the budget that I have and other considerations. For example, microscopy is definitely the gold standard. That is but you need the experts. You need the experts to do it. Sometimes it takes long period of time for you to really learn how to conduct an appropriate malaria microscopy. So if you don't have the expertise and you don't have an expert nearby, then from you are reading and analysis stage, you will know that microscopy may not be an appropriate method of a diagnosis for you. Then RDT, although quick and easy, but sometimes it couldn't differentiate the species that you are dealing with. So that is a defect for RGT. Sometimes you discover that PCR, for example, is the most sensitive and differential, but you may not have the budget to conduct PCR. So these are some of the things that during your design you will kind of optimize. And if you have enough of funding, you will see that all the three methods are actually complementary. You can do all the three methods, and they will give you useful information that you can include in your report. And then another consideration during your reading and analysis stage that you may discover is that sometimes what will decide what you do during your design is that you want to, after the study, one of the aims of researchers is to publish the results. So the findings of these results, where they are published, determines the quality of the publication, determines how you are going to be rewarded in the future in terms of grants, in terms of jobs, in terms of promotion and whatever. So that means that sometimes even during your design stage, you already start thinking about where are similar studies published. So so that you know that, because during the design stage, you are going to end up choosing one method and discarding another. You want to, by that stage, know where are studies that use only microscopy for diagnosis of malaria to arrive at prevalence, where are they published? That may be like, this is like the last stage. You have already optimized the method you want to use. Sometimes it's helped for you to think ahead when designing, like, the method I want to use, has it been published in good journals? Are good journals really interested in this kind of assay I'm going to do? Or this assay maybe has not been used for many, many years, then it's no longer useful, it's no longer publishable in quality journals, then I don't want to use it. So it's sometimes important. Then sometimes you may think about, what kind of conferences am I going to attend? So if you already have narrowed down that I want to attend a conference that is organized by, say, WHO on the sensitivity of RDTs, for example, or this company that produces RDTs, they do have a conference where they assess the performance of RDT all over the world, and there's a conference. So you may think about ahead that I want to design my work so that I attend this conference or that. So this is some of the consideration that after you have optimized, you can consider. What laboratories are doing similar work? This is something that you should already be planning. If you are doing your master's now or PhD, you should already be planning your future collaborations, because sometimes when you finish and become an independent scientist, one thing that will propel you forward is establishing good networks. So you need to start thinking, during my master's, where do I want to do my PhD? If I want to do my PhD in so lab, I need to have some expertise that they need. So you need to start thinking, in my master's, I really want to do microscopy, because I know that in my PhD, I want to attend Professor Tom Biden, for example, lab, and I know that he's really, really interested in microscopy. Then I will incorporate microscopy in my research, so that when I finish, and I will tell them that I actually did have idea on microscopy, and they will consider me. And what can sometimes the design also will involve you asking yourself, what future application do I have with the sample? So if you choose to do PCR, for example, then you know that the DNA you are going to extract for the PCR is going to be useful for you in the future. So this is another consideration that, okay, yes, then PCR is a good investment, because even if after I finish my research, I can keep the samples and I can use them for future collaboration or future research. So after you do all this reiteration stage, you will see that by the time you have narrowed down, you may have modified your topic. This is all during the design stage. You may have modified your topic to read something like, suppose you have chosen to use PCR or molecular diagnosis, then you may have to call it, should you name your topic into molecular prevalence of malaria among under 5 children in your state. So this is more specific, more direct, and will capture the essence of what you have done in your research. And that means that your paper will be, when during indexing and during search, will come up with people that are working in molecular prevalence of diseases. So this is all in design stage. So if the moment you send this topic to whoever is responsible, for example, in BioRTC, molecular prevalence of malaria among the 5 children, we already know that you have already done your research, you have already narrowed down to what you want to do. So what are some of the, you now at a stage where you start asking yourself, what kind of reagents do I need, for example? So you need to list all the reagents you need. I need a PCR master mix. I need agarose electrophoresis. For example, I need all those things, DNA extraction kit or DNA extraction method, the reagents that I need for DNA extraction. Or if, sometimes malaria can be diagnosed from whole blood, but this is for those that are working in the area. But what I'm just trying to say is, at this stage, that is when you start narrowing down to the actual reagents you need for your research. So now these reagents, for example, like master mix, like agarose electrophoresis reagents, DNA extraction kits, these are general reagents that not only people that are working on malaria, people that are working with cells, people that are working in other parasites, the people that are working with other biological tissues are also using these reagents. So it's sometimes you see that we do archive these reagents or stock these reagents for people that will be coming to the center. So these are some of the reagents that we ask if the center has. Do we have these? Then we say we have, because then these are not specific for a particular research. But for reagents that are specific for you, like primers, for example, if you are doing PCR on anything, primers are specific for your own research. So you need to ask yourself, this is something that may not necessarily, sometimes if the primers are common primers, somebody has used them in BioRTC and left them, we already are working in the area, you are collaborating with Dr. Mahmoud or Raouf, they already have the reagent or the primers, then you can use them. But in most cases, primers are specific for your own work. You are the one to design them. You are responsible for ensuring the integrity. You're responsible for ordering. We can give you contact for the people that will order, but then you are the one responsible for ensuring that you purchase them. So these are some of it's helpful for you to categorize your reagents into two: those that are common and those that are specific for you. And this is where a lot of the talk breaks whenever somebody wants to come. The moment we start talking and what are some of the specific reagents you need, and that is where people would, you see that somebody doesn't know the actual reagents they need. They just know that they want to run molecular work, but they have not really narrowed down and mapped out the reagents that they need for themselves, which not necessarily somebody in the center who is not working in your research may not know what primers are suitable for you for the diagnosis of, say, for profiling of bacteria species. If somebody is not working in bacteria, may not know which are the best, which work compared to which. So these are some of the things that you need to really know. The moment you have narrowed down to what reagents you need, another question you ask yourself is, what are the facilities do I need? So you need PCR machine, you need gel documentation system, you have listed them, and then you can send for the contact in BioRTC, do you have these machines that we can say, yes, we have these machines, or we don't have this machine, but you can use this machine to do what you want to do. Then other consideration that you can do during your design is you can find a facility like BioRTC that I want to go to BioRTC. BioRTC is doing molecular diagnosis, and they, for example, they are not doing karyotyping, for example, and you want to do karyotyping, both karyotyping and PCR, then you can find another facility that is doing karyotyping, come to BioRTC and do your PCR, and then you can go to another facility and do your karyotyping. So these are some of the things that I mean by you need to find the facilities that you want to do your research. And definitely you need to conduct cost analysis. Sometimes these things are very expensive, so you need to be asking yourself, can I afford? I know that a lot of Nigerian research, especially MSc and PhD students, do not have funding, so they usually are funding it from their pockets. So sometimes just because you see a work published by somebody doesn't mean that you can afford it. So you need to really conduct a cost analysis and ensure that whatever you are proposing, you can do it. I will give one more example before I close. I think I have, okay, this is about 20 minutes or so. So suppose you are working in pharmacology, your broad area is pharmacology, and you are interested in knowing the mechanism of actions of drugs. Suppose you have narrowed down, you want to assess the mechanism of cellular toxicity of acetaminophen. Acetaminophen is the active ingredient in Panadol. So you want to know what is suppose there are reports that it induces toxicity. So you want to know what are some of the mechanism of cellular toxicity of the drug. So you may ask yourself during your reading and analysis and taking notes, not taking stage, that I need cells. Cells are enough for me to, you may have now seen that some people have used animals, but you can also use cells. Sometimes if you can use cell lines, then you don't need to use animals because of the what I mentioned about the 3 R's: the reduce, refine, and replace of animal welfare. So if you can use cells, there's no point in you using a whole animal. So I can use cells to do that. So what kind of techniques do I need? For example, I can use metabolomics, I can use proteomics, I can use genomics, I can use fluorescence microscopy. So these are all the things you know your topic, you have asked your question, but you can use different methods to answer them. So you have narrowed down now that for example, I've narrowed down that I want to use, say, microscopy to assess to answer my research question. So I can now modify my topic to read something like an investigation into acetaminophen-induced morphological changes on HEFG2 cells. These are cells, liver cell lines, and if I can see what are some of the morphological changes that happen to them, I can use that as a proxy of the mechanism of action of of toxicity of acetaminophen. So you see now that this topic carries more information compared to just mechanism of cellular toxicity of acetaminophen. So this is this I have not even gone to the laboratory, but I know that I have excluded a lot of things. I've excluded that I'm not going to use metabolomics. I'm not going to use proteomics. I'm not going to use genomics. I'm going to use microscopy only to see what are some of the changes induced by to cells that are exposed to acetaminophen. So this topic is more informative. The moment you send it to anybody who wants to help you design your perform your experiment, they know exactly what you need, what you want to do. And then during this stage also, you may have narrowed down that you want to use fluorescence microscopy. Then you have to ask yourself, what kind of buffers do I need? What are some of the secondary antibodies? What are some of the, for example, DAPI stain, which is a general stain in any microscopy facility, fluorescence microscopy facility, you should find a DAPI stain or any other Hushed, for example, which is a DNA stain. So these are some of the common stains. So just because this is something that I also, during this you are reading, you may see that somebody uses DAPI to stain DNA. Somebody may have used Hushed. DAPI and Hushed are just different suppliers, but they are essentially the same function. They perform the same function. They bind DNA and they release fluorescence light that you can detect using fluorescence microscopy. So don't keep asking just because you've seen that somebody has published with DAPI and we tell you that no, we have pushed, and you just say, oh no, I need DAPI, and you go and purchase DAPI using which is expensive. So you have to think about what are the alternative reagents that are available when you are designing your work. What is this reagent and what does it do? That helps you to, that's why I say sometimes it takes long time for you to arrive at a methodology, because sometimes you need to read. And during your reading, you need to do a comparative or to read about what is the mechanism of action of any reagent you are going to use. It's very, very important that any reagent you are going to use, you know exactly what it does, because that will allow you to substitute. Like I just mentioned, DAPI and Hushed are dyes that can be substituted. So sometimes just because you see that the paper you want to follow doesn't use this stain, then you can't use it. It doesn't affect your experiment. It just stains DNA. So of course, you need to ask yourself, what are the reagents that are specific for me, which are, for example, primary antibodies if you are going to use fluorescence microscopy. So primary antibodies, they are specific for your own target. So you need to ask yourself, what are the primary antibodies that I need? Most likely, except if we are very, very young facility, so you find that we don't have a broad range of primary antibodies for people that have come to use and leave. So sometimes you see that you'll be the first one to use that particular primary antibody. So you need to think about, how do I acquire this primary antibody? Sometimes secondary antibodies are very common. For example, right now in the facility, if you come with your primary antibody, we do have secondary fluorescent antibodies that you can detect your, especially anti-goat and anti-mouse are available in the facility. So what facilities do I need? Because I'm using cells, I know that I need tissue culture facility. So that's, like I said, if you don't have tissue culture facility, then you can use animals, for example. But I also need fluorescent microscope, like confocal microscope or normal fluorescent microscope. So you also need to, like I said, find a facility and conduct cost analysis. Kundi, are you with me? Kundi,
Inshallah.
are you with me?
Yes, I'm with you. Yes.
Good. So that I just want to make sure. Okay. So please, those that are listening, if you have question, I think Kundi must have pasted it in the comment section that you can write your comments so that because I can't see your questions now, but definitely when I finish the sharing the screen, I should be able to see your questions and answer them. Okay, so how do you apply to visit by RTC? So suppose now you have finished, you know exactly what you want to do, your research. So you want to apply for by RTC, then you don't need to. It's very easy application process. We have an application form online. So you go to this BioRTC, www.biortc.com. You go to what we do, then you see lab visit application. You click on lab visit application, then it should take you to this page where to a form where you are going to fill in some key information. So I'm going to walk you through how you can fill this form because from those that have filled it before, I see that sometimes they struggle to understand some of the key parts. Of course, name, email, institute are something that you should have at the tip of your the fingertip. So I don't need to explain that. Position, sometimes we collect position so that we know what to expect. If you are a master student, for example, we know what to expect compared to when you are a PhD student, for example. If you are a researcher, we know what to expect compared to when you are, say, a technician. So this is important, please. What we mean here is, are you a master student, PhD student, or what are you doing? Are you a researcher, a seasonal researcher? Are you a principal investigator? What exactly are you? What is your area of specialty? The reason for that is sometimes for the area of specialty, when you write it, we are going to find sometimes if you have questions we have amongst the PI, if suppose you are proposing to collaborate, then we can link you up with the PI where you can discuss more in depth with them. Then you are going to tell us when you plan to come. So, like I said, please ensure that you plan to come to BioRTC when you have already designed your work, when you have already known the methods you are going to use, the machines you are going to use. There is no harm, like I said, to start asking questions about what the substitute of the things that we have unofficially. I mean, it's official, but using you can establish communication during your research, but just expect that don't expect an in-depth discussion to start to begin until you have filled this form. So you please ensure that you plan to come. Like I said, when you have already designed your work, you know exactly when you want to come. You are ready to begin. Then you fill the form. Because I do have some applications to come to visit the lab, but we are still waiting for them because they have not really narrowed down what they want to do, but they fill the form. So please ensure that by the time you want to come, you already have the plan, the date, your proposed date to come to the center. Then we will ask you for how long. This is just for a kind of planning purposes. We want to know our visitors because the center will be receiving a lot of visitors. So we want to know. Sometimes we don't know in the future. Sometimes there may not be available space for you. So if we have a lot of people there and you tell us that you want to spend 6 months, then we will plan that in the year 2023, second part of 2023, we have 20 visitors. Then we know that we can't accommodate anymore. Then we can close down the form. Now, what I want to really talk about is where tell us about your research and what experiments you would like to conduct at RTC. Please and please, here is not where you put all your research methodology as you have it in your research proposal. What we want here is, like I said in the research design that I gave you, is you have already finished everything. You know the experiments you want to perform, and you know the reagents you need, and you know the machines that you are going to work with. So please, this is the information we need, not your whole materials and methods could lump up together and say, I want to perform a FRAF assay as described by the 118 et al. No, you don't do that, please. Right? Please do have the actual methods, the actual experiments that you want to run. I want to run agarose gel electrophoresis of DNA. I want to run gel electrophoresis of RNA. I want to conduct XTS page electrophoresis followed by Western blot using the primary antibody against caspase-3 and the secondary antibody. If you already have the primary antibody, that the secondary antibody, goat anti-mouse or mouse donkey anti. What these are the data that we need so that exactly what you are going to do is what we want, please. Not your full materials and methods. Okay. Then we will ask you to agree that when you are accepted, you will be responsible for all the expenses like the transportation, accommodation, your upkeep. I'm going to talk more about lab consumables in my subsequent slide. And when you are during your period of stay in the center, you also agree, you must agree that when you come to BioRTC, because we are open access, you are going to use a lot of the resources there for free. We want you to acknowledge BioRTC in your future articles, papers, and theses that you are going to publish. And also, we have a set of rules and regulations that the moment we accept you to come to BioRTC, we are going to give you the link to these rules and regulations. You must agree that you are going to abide by the rules. And these are all rules that are general to any biomedical facility that you must ensure that you have by the safety. We are very, very serious about by safety. So you must ensure that safety. And if you damage anything, of course, because the lab needs to be sustained, then you have to agree that you are going to replace. Then you also have to provide a letter from your supervisor or head of department agreeing that you are going to visit by RTC and you have accepted this. So this is generally about the form. It's very easy to fill. You can fill it, and we will receive these responses, and we will initiate contact with you, and you can start preparing your visit to the RTC. So another question that I keep getting asked is, how much does it cost? Well, accessing the facility is free. We are an open access facility, meaning that the primary aim is to increase accessibility to facility to African scientists. So accessing the facility will not cost you anything. You don't have like a particular bench fee that you need to pay. But there is a but. The consumables are reagents you are bringing, like I said in the form, you will say that you are going to bring them, the consumables and all the reagents that you're going to use, you are going to bring your own. If you are going to use our consumables, these include tips, pipette tips, down to even hand gloves, our buffers, plates, and all these consumables that you will have to buy to replenish, then you are going to be charged for them. And for some machines that require maintenance, like the confocal microscope, like LICO, like CO2 incubator, these are machines that they are there, but they require you from time to time to spend money to maintain them because they have lamps that get damaged. Even if you naturally they are going to get damaged, you must replace them from time to time. CO2 incubator, you must buy the CO2. Liquid nitrogen, you must buy the liquid nitrogen. Chillers, for example, you need to buy the liquid nitrogen. So these are some of the things that, of course, we are going to charge you if you are using them because there are some of the things that need maintenance. And like I said, the facility, I explained how it's maintained. The equipment were received and they have been installed. Now the maintenance is on the center. So we need to keep charging. But by any means, the charges are not going to hinder you from your research because they are like the barest. They were set that with the bearing in mind that the students that are going to use them are going to pay from their pockets, most of them. So we are aware of that. We didn't like put exorbitant price for you. Then we'll give you free access to all our centrifuges, PCR apparatus, weighing balances. These are all the things, the common things that you find that you must pay in other laboratories. These are some of the things that when you come to BioRTC, you are going to use them for free. So of course, you now see that if you already have all your agents and everything, really BioRTC is a free facility for you to come and conduct your cutting edge research. So if you have I think I will close by saying that if you have any inquiry, if you can quickly jot down this email, like I said, if you want to initiate contact before you fill the application form, So please jot down this email and keep it in a safe place. Whenever you are ready to start to initiate contact with BioRTC, please you can send an email, and Kundi, who is the current technical manager in the lab, should contact you, should talk to you, and you can initiate contact and prepare for before you even fill the online form. We but RTC could not have been possible without the help of these institutions, associations, non-governmental and governmental organizations. Yobe State University, of course, provide funding to Yobe State University, and really, really helps in ensuring the activities on the center are running smoothly. Then you have Trend in Africa, who are coordinating the donation of the equipment and the receipt, and the whose the equipment are actually under the care of Trend. Then Charles Zuckerberg Initiative provided the funding to some of the PIs in the center for the running of the center. Then, of course, University of Sussex, then the Crick Institute that provide the equipment. Then Sussex Neuroscience that help in a lot of ways with funding and travel grants and seeing that the center is up and running. So these are some of the people that really we need to acknowledge. And when you come to visit the center and use the facility, you really need to also be aware that these are the organizations that make BioRTC possible and giving access to science to everyone in Nigeria and Africa at large. So I will welcome your questions. Thank you very much. Let me end the screen share.
Thank you very much, Dr. Bindawa. You have done more than justice to this topic. You have said a lot. A lot has been heard from Dr. Bindawa today. My question now, Dr. Bindawa, is you have talked science, Dr. Bindawa, but yet you have not talked about the environment. We know Yobe State is
Oh,
one
okay.
of the states in northeastern Nigeria
Yes.
where in the past we
Yes.
have some crisis. So, Dr. Bindawa, please, can you shed more light on how the security situation is in Damaturu right now?
Yes. I thought because I already introduced me that I'm not from Yobe State, I thought that would have been inferred that Yobe State is now. But I think I should mention it categorically. Really,
Yes.
when even myself, when I got the position in Yobe, because I wouldn't say myself personally because I did serve in Yobe State, so I don't have any problem going there. But a lot of people were really concerned about the stories they had, especially in the first part of the last decade. But I'm telling, this is something that is very, very probable that the Bodamatru is one of the safest states now, in especially in northeastern Nigeria. In northern Nigeria, Bodamatru is one of the safest states. Have a very, very good road access from Kano. You can cruise if you have a car that can cruise. You can cruise to Damaturu without having to dodge any potholes or anything. So I think I would comment that I myself am not from Damaturu, and I spent about 8 months there, and I'm going back in the next couple of weeks. So the security situation really improved, and this is something that you shouldn't worry. And in the last summer school, we had international faculty, more than 10 international faculty that came, and we didn't have. They were really, really impressed with the security situation and the services they received during their visit to Damaturu. And I assure you, if you are coming to the RTC, this is something that you also can expect. The hospitality, the peace, and the tranquility that is in the state is really, really commendable. Yeah. So I thought it was inferred, but glad you reminded me that I have to say it categorically that I'm really impressed with what is happening in Damaturu. It's only that the temperature there gets very hot. So you have to prepare
What
when you are
about
going.
people coming from Borno?
Yes. So when you're going there, ensure that you take you plan to take light clothes, especially during month of March up to maybe I say September, October. Yeah. Do we have questions in the comment section?
In the comment section, there is no much question. I guess there is no even a single question here. Most of the comments were on appreciation. They appreciate the talk. Thank you, Dr. Muchala, for wonderful presentation. Question is okay. Somebody is asking: is there any funding aid in BioRTC?
Is there any funding? So I think okay, let me see. Can we share for those researchers that are working on in bioimaging? If you are using imaging in your research, currently African Bioimaging Network, I think in collaboration with CZI, funding visitors. If you already have a research ongoing that involves microscopy, you can apply. BioRTC is one of the selected centers across Africa. This is not only in Nigeria, across Africa. Of course, BioRTC is the only center from Africa that is selected. So you can apply for the grant. I think what we can do is, I don't have the link now, but it's Dr. Mahmoud available. Anyway, we are going to share. I think what we can do is share across the platforms that we shared the webinar invitation for the webinar. We can share the link for the grant. So you're already working, if you're already working with in microscopy, you can apply for the grant to visit and spend some time in BioRTC to conduct your research. It's a very good, up to $2,000 for you to visit BioRTC for your research. But if you are talking about BioRTC funding where we have money for you to come and do your research in the future, but not now, really.
Yeah. So, Dr. Mutsala, here there is Muhammad Ado Musa and Hidayat Jimo Abdul Ghaffar. It seems like they were lost. They were asking a question that is very relevant to what you've been discussing during the course of the talk. Muhammad Ado Musa was asking whether my question is how can somebody be able to conduct his or her research in the RTC, and what kind of maximum support one can get when doing his research work? You see, you've discussed this.
Yeah, I think I discussed it. So the recording of the
Yeah.
webinar is going to be available on the website, so you can go and
Yes.
watch it, and you should
Yes.
be able to get it from there.
And Hidayat, who is one of our fellow who attended summer school, the first summer school, she was asking whether there is a provision for accommodation for the
For
PG
PG
students,
students.
for the researchers.
Yeah, so there should be provision, and this is one thing that I said you can initiate contact before you come. So Kundi and Kulayas with Abdurrahman, maybe you can create
Yeah.
a comprehensive guideline on how to access the accommodation available
Okay.
for the center. Yes,
Yeah.
I think we are over
So
time. If we
yeah,
can,
it's almost an hour. So thank you everyone for attending.
so
See you
you
guys in our next webinar series. Dr. Muchola.
Okay, thank you very much, everyone. I'm looking forward to see you in BioRTC, in future BioRTC activities. And like I said, if you are working in, especially neuroscience, you can apply for the 2023 summer school, which is the application is ongoing, which is going to hold on in August. Thank you very much. Bye-bye. Thank you.
Bye everyone.